So girls, I will continue with the structure of a seed. Okay, how the seed will form. We have studied yesterday the formation of the embryo. Now this embryo, it will mature and finally form the seed. So we will study the structure of a seed. Okay, the at maturity, this ovule, as you can see here in the female cone in the figure A, the ovule is finally transformed into a seed. And I had mentioned before also that the seed, as you can see, it is oval in shape, and also the color of the seed is usually green to red. Okay, green to red in color. And this fertilized ovule, it undergoes the following changes, as you can see in these two figures B and C. The figure B is the transverse section when you cut the transverse section of a seed, and figure C, it shows when you cut the longitudinal section of a seed. So what will happen to these actually? Here you can see there is the outer layer, the middle layer, and the inner layer. But as the seed mature, what will happen to the outermost layer? Just listen. The outermost layer of the ovule, it will persist in the seed. Okay, it remains as it is, and it will provide a green to red color to the seed. This layer, that is the outer layer here, is parenchymatous, and also it is fleshy, but it remains free from the base to the apex of the seed. As you can see in figure C as well as B, it remains free after the maturity of the seed. Also, it remains free. It will not attach or merge with any layer. Now the middle layer here is actually the outer integument, right? It has become the middle layer of the seed, and this middle layer, it will actually grow above the outer layer. Okay, when the seed it become mature, the middle layer will actually grow above the outer layer of the envelope, and it will surrounds the micropylar region, the micropylar tube, and it will form the hard protective covering. Okay, it will form the hard protective covering of the seed. And also this layer, the middle layer, after it forms the hard protective covering, it is differentiated into palisade, sclerenchyma, as well as parenchymatous zone. And also many vascular bundles are found in this layer. Coming to the inner layer here, or the inner integument, it remains fused with the nucellus up to 2/3 of its length. Okay, you can see here the nucellus. It remains fused with the inner layer, and the free portion of the inner layer, it encircles the nucellar cap, and it will project beyond the nucellus. Okay, where the you can see here just below the inner layer, the nucellus is present, right? So the nucellar, this inner layer, it encircles the nucellar cap, and it will project beyond the nucellus. Now the nucellus will become thin, and it remains in the form of a thin strip around the upper part of the endosperm, while the endosperm, it remains fleshy and becomes the kernel of the seed. Okay, this endosperm, it will become the kernel of the seed, and the center part of the seed is occupied by single dicotyledonous embryo, as you can see here in figure C clearly, which has persistent feeder. Okay, you can see here the feeder is present in the middle portion of the endosperm. Now let us take a look how this seed will germinate. So now the seed of Gnetum, they are shed from the plant. Okay, after they become mature, they will shed from the plant the seed, and while the embryo is still developing. Usually they shed after the formation of the secondary suspensor, as you have already studied, and further development of the embryo, it will takes place in the on the ground itself when they are shed. Therefore the seeds, they do not germinate immediately, but they require a certain period of maturation. Unlike the seeds of pinus and cycas, where we have seen that as soon as they reach the ground, if the favorable under the favorable condition, they start to germinate. But the Gnetum, the seeds, they do not germinate immediately. Okay, they require a certain period for maturation. And for example, one of the species of Gnetum ula, okay, Gnetum ula, they may remain extended for a period of up to 11-12 months. After that only they will start to germinate, but it depends from species to species. Now a fully ripened seed, it absorbs water and it will swell. The radicle will come out first and it will penetrate into the soil, as you can see in figure A. Then the hypocotyl, it will elongate, pushing the two cotyledons into the air. You can see in figure B and C. And the plumule, it will grow out and form the shoot system. The cotyledons, they become green and photosynthetic. Okay, you can see these which leads to the formation of the leaf. Whereas the feeder, it remains inside the seed and it will continue to absorb food material. So that is the function of feeder. Okay, you can see in figure E. This feeder, it remains inside the seed and it will continue to absorb food material from the endosperm till the young shoot, it develops 2 to 3 pair of leaves. So this is the whole process of the germination of seed. It is similar in other gymnosperm, as you have studied the cycas and pinus as well, but just remember that in case of Gnetum, the seed, they do not germinate immediately but require a certain period for maturation.